Life is something that is extremely abstract to talk about, whether it be its purpose, what qualifies as life, or even if there is life. This is also very true when discussing the origins of life, as many of the theories proposed as to the beginnings of our existence are difficult or near impossible to prove, or are simply too unlikely to have occurred. At our current progress, the most consistent theory that we have is the RNA world theory, which describes how life originated from an RNA molecule that could self-replicate, eventually leading to more complex forms of cellular life. The theory is evidenced by RNA's various chemical properties and functions in life, giving us an idea of how RNA can be used in a different setting to create our own forms of life in future scientific advancements. However, it is still limited, as scientists at present are unable to fully understand or recreate certain aspects of this theory. Nevertheless, it is still the most important and relevant theory for the origin of life that modern science can manage. The journal article we chose to reference and use as a baseline for this presentation was published on PNAS by Patrick Fortier and concerns the origin of different cellular domains. Although it does not specifically refer to the RNA world theory, it indirectly explains many of its key components and how it would go on to create more complex life.
Although there are several theories as to how life originated on Earth, there are still missing pieces of the puzzle, such as understanding the atmosphere and environment of primitive Earth. An atmosphere containing free oxygen would be fatal to all origin of life theories. While oxygen is necessary for life, such as the ozone layer, free oxygen would oxidize and thus destroy all organic molecules that are needed for the origin of life. With this much evidence that the Earth requires oxygen, there are theories that say that there was no oxygen in Earth's early atmosphere. However, this would be fatal to an evolutionary origin of life. If there were no oxygen, there would be no ozone layer around the Earth. Not including all the other important factors of the ozone layer, if there was no ozone, the destructive, unfiltered, ultraviolet rays of light from the sun would shine down on the surface of the Earth, destroying all organic molecules required for life, reducing to simple gas such as nitrogen, carbon dioxide, and water vapor. Thus falling into the predicament that in the presence of oxygen, life could not evolve, and without oxygen, life could not evolve or exist. The energy available on the hypothetical primitive Earth would consist mainly of radiation from the sun and other destructive energy sources such as lightning. The problem for creation of new molecules and such is that the rate of destruction of these energy sources surpasses the rate of formation from the same energy source. In Miller's experiment to create life, the products were isolated from the energy source. There would be no such mechanism to trap and isolate the products in early Earth. And if there was a mechanism to trap and isolate these products, there would be no further evolution, as energy is needed from these sources to continue evolution. The UV light more specifically affects the ability of DNA and RNA to survive, as the UV light breaks down bonds of the bases forming photodimeric liaisons, as shown in Figure 1, which inhibits transcription and replication of these polymers. Which brings up the question, how could DNA and RNA exist in such brutal conditions? Sadly, we are unable to answer these questions due to our lack of understanding of the origin of life, as not much is known about early Earth and the ability to arise life from non-living matter.
The RNA world theory is the idea that in the times of around 4 billion years ago, which was the early stages of life, life consisted primarily of RNA, and that RNA did most of the metabolic processes and the information processing, and that RNA was the reason life was able to emerge into much more complicated organisms. In this theory, it is said that RNAs were formed through a chaotic soup with millions of molecules. As nucleotide was bonded, they formed RNA strands, although they would break down soon after. Eventually, as time passed on, the RNA strands became more and more stable, as the RNA strand grew faster than the speed at which it was breaking down. This finally allowed the RNA strands to evolve as they started competing with each other, the most adaptive and stable RNA coming out as the main RNA strands to bring life to the world. Eventually, these RNA strands began to replicate as they copied their genetic material and passed it on. Again, the RNA strands with the best survivability were the main RNA strands to bring more life to the world, as the weaker RNA strands were filtered out through natural selection. As RNA evolved, proteins and DNA came about. As DNA is more stable than RNA, DNA then took the job of safely storing the genetic information. Then, through billions of years of evolution, here we are today.
Over time, much evidence for the validity of this RNA world theory has been discovered. As pointed out by Garland Science, the formation of cellular life requires autocatalytic processes to produce more versions of itself. If RNA were the first source of life, it could have coded for the polypeptides that provided these specific processes in more advanced prokaryotic cells. Additionally, RNA is an extremely versatile molecule. As this diagram shows, its complementary base pairing makes it able to be used to bind to more complex molecules or to code for more proteins and amino acids. Therefore, it seems plausible that the presence of RNA in the primordial soup was able to give rise to other compounds that would therefore form complex life. This is supported by Patrick Fortier as well, who suggests that subsequent RNA viruses could have been given rise to RNA transfer and cell formation. Additionally, studies by Tohoku University have found samples of ribosugar and phosphate in meteorites formed at the same time as the Earth, indicating these compounds were indeed present at the time of life's formation. Either they also formed on Earth or were carried to it by meteorites, proving that RNA could have formed entirely in the primordial soup as well. As shown from these properties and conditions, they are entirely supportive of the process of primordial RNA giving rise to more complex forms of life we see today. However, our lack of ability to recreate the conditions of primordial Earth does not give us specific information about how such RNA functions would actually proceed in creating new life. The Miller-Urey experiment produced sugars and phosphate, but could not produce any RNA, for instance. We are still quite limited in what we can see in terms of evidence of the RNA world theory.
What are the challenges of the RNA world hypothesis? Although the evidence of RNA world hypothesis is still strong, but there are a few significant challenges, such as proving prebiotic conditions and its ability to spontaneously create complex polymer, such as RNA. First, RNA is a large molecule made of a long chain of nucleotide, which contains 4 nitrogen bases, sugar and phosphate groups. Abiotic nature has a bias towards the formation of molecules made of fewer numbers of atoms, thus shows not in favor in creating the complex building blocks of RNA. Secondly, there is also a problem of classifying prebiotic conditions, because a prebiotic condition itself is another hypothesis. With John Sutherland's experiment, for example, pyrimidine ribonucleotides, which polymerize to form RNA, were synthesized in plausible prebiotic conditions. The important feature of this synthesis is the multiple roles that inorganic phosphates have in the overall reaction. However, the availability of inorganic phosphate in prebiotic conditions is still scientifically not proven. Robert Shapiro criticizes that the prebiotic conditions were conveniently created to support RNA world hypothesis, because once a specific organic chemical is classified as prebiotic, then any product of reaction of this chemical can then also be considered as a prebiotic molecule. There have been many suggestions in terms of early Earth features to support the correct prebiotic conditions to suit their studies, such as drying lagoons, glacial lakes, and so on. But there is not a feature that exists on the Earth today that gives us definite answers to it.
From this, we can conclude that answering the question of the origin of life through the RNA world theory is entirely possible, but it's still very limited in the scope of modern technology. With future technological advancements and funding, the RNA world theory could be completely incorrect, or the answer to everything about life and history. But in this modern era of science, it is the best framework that we have, and a perfect scaffolding for building and connecting new theories surrounding the origin of life. It is entirely important for the future advancement of science and prehistoric research as we know it. Thank you so much for listening, and have a lovely day.